Hello, and a very warm welcome to everyone joining us for today's Select Science webinar in partnership with SPT Lab Tech, titled How to Accelerate Small Molecule Lead Discovery with Automated Multi-Channel Pipetting and DEL Technology. My name is Blake Foreman, and I'll be moderating today's presentation. I'm delighted to be joined by our guest speakers, Zoe Wang, Research Scientist, and Dr. Paige Dixon, Principal Research Scientist at XChem. In this webinar, we will cover how to establish an efficient compound testing workflow using the Mosquito LV. We will discuss the benefits and considerations of a robust biochemical assay, as well as an overview of the use of DNA-encoded library technology as a small molecule hit discovery approach. Following the presentation, we will have time for a question and answer session. If you have any questions, please feel free to submit these at any time during the webinar. You can submit your questions to the left of your screen. Simply hover over the questions icon and click to type in your question. Without further delay, I'd like to hand over to our speakers, and I'd like to thank them again for presenting for us today.
Hi, everyone. Thanks for attending today's webinar, where we'll focus on multi-channel pipetting and DNA-encoded library technology, and how these techniques together can accelerate small molecule lead discovery. My name is Paige Dixon, and I'm a Principal Research Scientist at XChem. I'm here with my colleague Zoe Wang, where we'll be focusing on these topics as we go through the webinar. As an outline for today's discussion, first we'll start with early-stage small molecule drug discovery overview, discussing the different phases of this process and the roles that XChem has within this workflow. Next, we'll discuss how Mosquito LV aids in the validation of hits derived from a DNA-encoded library screen. And lastly, apply these workflows to a case study of HAO1, a diseased target protein which XChem has studied and published on in the past. First, as an introduction to early-stage drug discovery, we see several stages leading up to candidate selection in the downstream clinical trial work. XChem's primary focus is on these first three stages of early-stage drug discovery. First, targets must be identified and validated, typically pursued through genetics, molecular biology techniques, and cell-based and animal-based studies. Once suitable targets have been identified for a particular disease, small molecule or biologics must be identified towards those disease targets of interest. At XChem, we focus on a particular screening modality known as DNA-encoded library technology. However, there are a number of other hit-finding modalities, including HGS and fragment-based drug discovery. Once these hits are identified, a series of different validation assays will be performed to profile the pharmacology of the hits, their basic properties in vitro, as well as general physicochemical behaviors. With these initial hits in hand, medicinal chemistry will allow for iterative optimization, generating a lead molecule. Once a lead has been identified, further optimization will be performed to improve the in vitro and in vivo ADME properties and start to allow for initial animal efficacy studies. Here at XChem, our focus is around these early stage processes in small molecule drug discovery. And our mission is to deploy our world-leading drug discovery platform to help our partners create molecules that improve health. And the ways that we do this are through our three pillars of expertise, focused on DNA-encoded library screening to identify novel small molecules towards disease targets, medicinal chemistry to support the development of screening hits into tractable leads and ultimately clinical candidates, and lastly, artificial intelligence to support DEL screening, hit finding, medicinal chemistry, and the entire drug discovery process. And here at XChem, we have a world-leading suite of predictive and generative models to accelerate these drug discovery workflows. At XChem, our passion for hit finding is based on DNA-encoded library technology, and we believe that this approach is highly valuable for efficient and resource-mindful discovery of novel ligands. Traditional high-throughput screening is often limited by the linear correlation between the resources required and the size of the compound library that can be screened. Because each of the molecules within an HTS deck are physically separated into wells of a multi-well plate, that means for each additional compound that is within the collection, there's an increased amount of resources in terms of assay reagents, protein reagents, and time to prosecute these additional compounds. DEL is particularly advantageous for screening large library collections because the entire library can be screened in a single tube, allowing for billions of members of the library to be interrogated simultaneously. And as opposed to traditional HTS, where large suites of robotics, expensive equipment, and a very large lab footprint is required to prosecute these millions of member libraries, DEL screening can be performed in a very small footprint using benchtop robotics such as this SPT Lab Tech Firefly. With that introduction to drug discovery, I'd like to introduce DNA-encoded library screening a bit more before transitioning into our case study. DNA-encoded libraries are generated through a unique synthesis process known as split and pool chemistry. The DNA-encoded library architecture is divided into two parts. The small molecule component, which is the chemical entity that we're trying to screen within this experiment. Attached to each of these small molecules within our library is a unique DNA barcode, which we can use to sequence the identity of our hits that come out of the DEL screen. DNA-encoded libraries are particularly advantageous because we can generate millions, if not billions, of compounds with very few synthetic steps and incorporating a large diversity of chemical equity within these libraries. Here at XChem, we have over 100 billion compounds in our current library collection, and our libraries reflect both covalent and non-covalent chemical equity. With these libraries, we can take an affinity tagged target of interest and incubate the libraries along with this target to facilitate binding events. Once binding events between our small molecule library members and the protein targets have occurred, we can immobilize that protein via the affinity tag, wash away unbound library members, and elute with heat in a very similar process to a Cohen immunoprecipitation experiment. This process will be performed 2 times to successfully enrich library compounds, which we can detect through PCR amplification and subsequent DNA sequencing. A typical workflow for a DEL screen performed here at XChem is illustrated on this slide. With our library compounds in the high billions of range, we can enrich for affinity binders in the process I described on the previous slide, and then characterize the identity of those using deep sequencing. Once we have these sequences corresponding to our enriched molecules, we can translate those sequences back to the chemical structures that they were associated with, and through statistical analysis, identify the hits that came out of the screen. Because a DNA-encoded library contains billions of members, we group the hits that come out of the screen based on their structural similarity. And this allows us to more efficiently prosecute those hits with off-DNA synthesis and subsequent validation in a traditional biochemical or biophysical assay. And with that, I would like to hand it over to Zoe to describe how the mosquito can support our assay testing workflows.
Great, thank you, Paige. As Paige mentioned, once we have identified compounds through DEL selection, our next step is to utilize assay for screening those binders. In this phase, a robust and efficient assay workflow is very important because this assay result will significantly influence the direction of our next step. In the upcoming slides, we will provide a brief overview of our standard compound testing workflow, and we will also dive into how the mosquito system enhance and integrate in our assay process. Great. This slide here shows our standard 3-phase compound testing progress. The phase 1 is the assay development. We first need to establish assay by determining the optimal concentration of the reagent in the assay. As we can see in the green box here, it shows a protein titration data example to find the linear reaction range. We also validate the assay by using control compounds for the target that has known binders. After we establish assay, we have 2-step compound screening process. The first step is to testing compounds in the 3 doses. As we can see in the blue box here, each dose have 3 independent duplicates. This data can help us to see if the compound is active and if the compound has a reasonable dose response. In the phase 3, we select the actives from 3 dose tests and test them in the full dose. As we can see from the assay 50 curve in the pink box, each compound is tested in the 10 doses with 3-fold dilution, and each dose has 2 duplicates. In our next slide, we will explore how the integration of mosquito technology help in mapping those compounds efficiently across the 3 doses and the full doses compound plate. Right, let's take a closer look at these 2 plates we have up here. Starting with the top plate, which is our example of the 3-dose plate map. The column in the color mint and purple are the positive control and the negative control respectively, and each square in one color indicates one compound. For example, from the column 1 to column 3 is from high concentration to the low concentration, and from row 1 to row C are the 3 independent duplicates. The full dose plate map shows in the bottom here. The column in the dark purple and green are the positive control and the negative control respectively. And again, each section in one color indicates one compound. From column 1 to column 10 is the 10 doses 3 dilution for one compound, and row A, row B are the independent duplicates. So how exactly does mosquito help us to mapping those plates? We will use one full dose plate here as an example to look into detail. In the blue box here, it displays a half plate example for the full dose plate map. The plate 1 is a stock plate that we usually make manually. Column 1 has the 70 microliter of the DMSO for compound dilution, and column 2 has 8 compounds. You can imagine that each 2 wells in one color is one compound in duplicate at the top concentration. Plate 2 to plate 5 here shows the compound plate status under each step. At the very beginning, mosquito will first transfer 1 microliter DMSO from the stock plate to compound plate column 11 and column 12 for the positive control and the negative control. After that, mosquito will transfer 1 microliter DMSO from stock plate to the compound plate 2 for the 2 to compound plate column 2 to 10 for the 3x dilution. After that, 1 microliter of the compound from the stock plate column 2 will be transferred to the compound plate column 1. And in the step 5, mosquito will repeatedly take 500 nanoliter solution from previous column into the next column to achieve the 3x zero dilution. And you can also imagine for the second half of the plate, it just simply use the compound solution located in the stock plate column 3 and repeat the step 2 to 5 for the second half of the plate. After compound plate is being made, mosquito will help to copy the compound plate to the assay plate, and the compounds are ready to be tested. That's pretty much the role of the mosquito in our compound screening process. Coming up next, Paige and I are going to dive into a real project case study. We will show you how DAO selection works in a real-world project and how the mosquito fit into our whole process.
Thanks, Zoe. Great. Jumping into the introduction to our diseased target, HAO1. This protein is implicated in a rare liver disorder where glyoxylate metabolism is dysregulated. Patients which carry this rare liver disorder, PH1, have increased levels of oxalate, which can lead to progressive kidney deterioration. HAO1 is one enzyme within this metabolic pathway, and it is envisioned that inhibiting this protein is a strategy to mitigate the accumulation of toxic oxalate in PH1. Thus, we generated this protein recombinantly to support a DNA-encoded library screen. Before performing a DEL screen, protein quality control is extremely important to ensure that the species we're screening is our desired target of interest. XChem has several different DEL screening offerings, and the HAO1 protein was screened with our DEL core offering, which contains a non-covalent library of over 100 billion compounds. Before we describe the results from the DEL screen, I'd like to introduce some of the protein quality control activities that we perform here at XChem. First, we perform a DEL-specific interrogation of the protein's capture and activity in its immobilized state. This experiment allows us to determine whether the protein is able to associate with the desired affinity resin and whether that protein, when immobilized, is active. Often we adapt an existing in solution assay to this on-tip experiment, and this experiment is no exception. So in this workflow, we prepare our protein in our selection buffer, and we save a sample for input analysis on SDS page. We then flow the protein over the affinity resin for 30 minutes, allowing for that tag to interact with the solid support. We save the supernatant as the flow-through for our SDS page analysis, and in an ideal case, there will be very little, if any, protein in this flow-through. Once the protein has been immobilized, we can flow the substrate mix for that particular activity assay over the tip. In this case, because we're looking for HAO1 turnover, we include the substrate glycolic acid, as well as two secondary detection reagents, Amplex Red and HRP. Zoe will describe this assay workflow in a later slide, but needless to say, by flowing this mixture over the tips, we're able to interrogate HAO1 activity in a fluorescent assay. And by collecting time points ranging from 0 to 15 minutes, we can measure the time-dependent accumulation of fluorescence as a readout of enzymatic activity. And indeed, we can see two different concentrations of immobilized HAO1 showing time-dependent increase in fluorescence as compared to a no target control, which is just a tip lacking protein, which has seen this substrate mix. When we look at the SDS page gel to assess capture efficiency, we can see that the two different concentrations of protein are reflected in the intensity of the bands in the input sample. What is nice to see is that there's very little protein in the flow-through, suggesting this capture efficiency is extremely robust, and there's almost stoichiometric recovery of the protein on the matrix sample. So this tells us that the protein captures very well to the affinity resin, and it isn't active when immobilized. In addition to these DEL-specific quality control experiments, we also perform traditional biophysical characterization of the sample and solution. To do so, we perform dynamic light scattering to measure the presence of aggregates across the sample. And for this HAO1 sample, we see the entirety of the mass present at a radius corresponding to the expected molecular weight. We do not observe any high radii sample particles, which would be indicative of aggregation. Furthermore, we characterized the molecular weight of the protein by size exclusion chromatography relative to a gel filtration standard. And we observed that the protein is indeed a functional multimer as based on size exclusion chromatography, which is expected based on what we know about this protein. With these quality control results in hand, we proceeded to enrich for affinity binders to HAO1, characterize these using sequencing, and then identifying the enriched compounds through statistical analysis. In the interest of time today, we won't describe all of these steps of the workflow, but I encourage you to look at our publication for more information. Once we've synthesized these compounds off DNA, we're able to proceed into the assay testing phase of the DEL-core workflow. And with this, I'll hand it back over to Zoe to talk through the assay testing process.
Thank you, Paige. This slide here shows our high-level compound screening strategy. Besides the DEL selection process that Paige just described, our machine learning team also used the DEL selection data to train the model in order to identify the novel inhibitors of HAO1. After the new binders were found, our medicinal chemistry team also explored the hits by using methods such as SAR. All those compounds were tested by our primary assay, HAO1 HRP coupled biochemical assay. The assay principle can be found in the left corner here. HAO1 first converts the glycolate to glycoxylate. In the meanwhile, oxygen is transferred to the hydrogen peroxide. After that, HRP transferred ambidextride to the fluorescent version, and we use this signal to estimate the activity of the protein. Since we introduced the second protein HRP into the assay system, a HRP selective biochemical assay, selectivity biochemical assay is also established to filter out any false positive that actually bind to the HRP but not HO1. Additionally, a thermal shift assay and a orthogonal biochemical assay also be established to confirm the actives from the primary assay. We use this assay strategy to test nearly 1,000 compounds, and among those 1,000 compounds, we screenshot some of the compound results today to show how mosquito help us to produce accurate and reliable results. As we can see, the three figure here, the figure A shows the 4 active compound result. As we can see in the second column here, their IC50 are in the range of 20 nanomolar and 500 nanomolar. For the compound 3 and the compound 5, we got a pretty low standard deviation across 100 and to 150 repeats. That really shows how reproducible our process is. And the figure B here is for IC50 curve example. As we mentioned earlier, we test each dose in duplicates. We can see from those curves, all doses duplicates has relatively narrow error bars, which demonstrates the consistency of our process. The last figure, figure 3 here, is the compound testing result for one compound, but they are testing in winter and summer respectively. As we can see, the IC50s are pretty consistent between each other. This example really proves the reliability and the reproducibility of our process under different conditions of time, temperature, and humidity. Another interesting comparison we show up here is a comparison between the different assays. As we can see from the top graph here, is a comparison between the orthogonal assay result and our primary HO1 HRP assay. It shows very strong correlation when IC50 is below 5 microliter in the HRP assay. Although the IC50 is in the orthogonal assay is less potent compared to the HO1 HRP assay, we can still see the very clear linear correlation between these two assays. On the bottom graph here is a comparison between the orthogonal assays and the thermal shift assay. We also see a nice correlation between the enzymatic assay and the binding assay. This result further enhanced our confidence in the compound testing result and our whole assay workflow. All right, after screening nearly 1,000 compounds, it's time to show some of our most potent inhibitor that we found in this project. I will transfer back to Paige to introduce those vendors.
Great. So as Zoe mentioned, here are 3 of the hit structures that were derived from our HAO1 screen. And I'd like to point out that these 3 different hit series are derived from distinct DNA-encoded chemical libraries. So while some of the building blocks may overlap within these libraries, the chemical schemes that were used to generate them are quite distinct. So we can see here in DNA-encoded chemical library A, we have this core amide with various different building blocks coming off of the oxygen portion as well as the nitrogen portion of that amide. In DNA-encoded chemical library B, 2 different building blocks were attached to that core nitrogen. And in DNA-encoded chemical library C, a heterocyclic core is present, affording a very unique scaffold that is distinct from libraries A and B. What we can see here is that across these 3 hits, there are a variety of different physicochemical properties, but all of these are well accepted within the rule of 5 space. We also see some attractive SAR across these different series, which gives us good confidence that these molecules are truly interacting with our target. We can see from the compound of library B and the compound from library C, there's a very similar bicyclic building block that is present, which is likely making similar contacts with the protein in both cases. Our most potent hit came from DNA-encoded chemical library C. We were able to successfully characterize this not only with the assays that Zoe described previously, but also with further cell-based work. We were able to generate a co-crystal structure of this ligand with HAO1 protein, revealing a very unique binding mode. And we also observed through some in vivo studies that the mean plasma concentration of this protein is quite stable across a 24-hour time course. So in addition to the in vitro binding data we were able to elucidate and confirming the inhibition, we've also demonstrated that these compounds are potentially progressible for that PH1 disease. And HAO1 is not the only example of a successful DEL screen that XChem has reported. We have over 16 published examples of successful screening at this point, but 16 of those are shown here on this slide. And of these successful DEL screens we've prosecuted for our clients, we currently have 5 clinical candidates which are being explored as a result of this, the equity derived from the DEL screens. I would like to point out that across the different publications that we have, there are a number of different target classes across enzymes, kinases, protein-protein interaction mechanisms, as well as a variety of other species for which we've identified unique chemical equity towards. And this has covered a number of different therapeutic areas, including rare disease such as HAO1, as well as oncology, neurology, and infectious disease as well. And I think this slide really illustrates the unique chemistries that are derived from the DEL screen. We see the HAO1 inhibitor that I showed on the previous slide in this top row. We have a number of other compounds that really represent very distinct chemistries, which are reflective of the diversity within our library. I'd also like to highlight the one published example we have of a covalent inhibitor, which is a very unique mechanism that is emerging of interest for early stage drug discoverers. And so with that, I hope today we've convinced you that SPT lab tech liquid handlers, such as Mosquito LV, greatly enable high throughput compound screening to accelerate DEL workflows and early stage ligand discovery efforts. And I also hope we've conveyed that DEL technology is a powerful approach for identifying novel ligands for small molecule drug discovery programs. And I'd like to take the last few minutes to just promote several future directions we could envision for high throughput pipetting in our DEL discovery workflows. First, XChem uses machine learning to support our DEL screening processes, and the result of this machine learning workflow is more compounds to test. And so one could imagine that integrating this compound testing workflow of machine learning derived hits to supplement our traditional hit finding approaches would further accelerate this screening process. It also helps us to maximize the data that comes out of the DEL screen to fully enable hit finding for de novo ligand discovery. Another application of mosquito and high throughput liquid handling could be through the synthesis and preparation of DNA-encoded libraries for screening. And so since DNA-encoded libraries are synthesized through split and pool processes, where hundreds, if not thousands, of chemical reactions proceed in parallel, one could imagine that the liquid transferring capabilities of the mosquito would support these kind of split and pool synthesis processes. And further, since we end up mixing all of our libraries together to perform our DEL screen, one could imagine similarly that the mosquito can support the combination of libraries once we've completed this synthesis process. And with that, I'd like to thank you all for your time. To provide this resource for additional information if you're curious about any of XChem's capabilities for your hit finding projects. And I'd like to thank you all and open the floor to questions.
Thank you for that very informative presentation. It's now time for our question and answer session. So if you have any further questions, please do send those in. You can submit these by simply typing them into the questions icon to the left of your screen. You will also shortly see a poll question appear on your screen. Please feel free to answer this at your convenience, and we look forward to seeing your responses. So our first question for today is, what are the typical volumes you use in your compound testing workflow?
I think I can take that question on. So typically we perform ultra low volume reactions for our assay testing. This is not only to save reagents, but also to maximize the throughput of those experiments. So a typical volume of compound that we would dispense is 100 nanoliters of a DMSO stock. However, the mosquito can handle volumes below 100 nanoliters. We've gone as low as 40 nanoliters. It can also go up to a whole microliter or 1,000 nanoliters. So depending on kind of the assay capacity as well as what has been established for that particular project, this may range from 40 nanoliters all the way up to 1,000 nanoliters.
That's great. Thank you for that answer. And our next question is, not all compounds are dissolved in DMSO. Have you ever used other solvents with this workflow?
Absolutely. So our traditional solvent is indeed DMSO. However, we have had projects where dispensing aqueous-based solvents, either pure water or buffered solutions, have been successful with the mosquito. We can also handle a variety of other solvents using the system. However, the viscosity of those solvents would have to be taken into consideration when developing those dispensation protocols.
Perfect. And just a reminder to our audience that if you do have any further questions, please do send those in. You can submit these to the left of your screen. Simply hover over the questions icon and click to type in your question. So we'll move on to our next question, which is, what is the typical compound number tested from the output of one DEL selection experiment?
Yeah, thank you. I can take this question. There are actually multiple factors that would affect the testing number for the DEL selection. Depending on the budget, we usually recommend synthesis 20 compounds for compound testing. But for a very productive DEL screen, the number might be like 50 to 100 compounds.
Great, that's an excellent answer. And our next question is, what other assay formats have you used to generate compound testing plates with the mosquito?
So the full dose and the 3 doses compound plate that we introduced in our presentation are more standard plate format that we're using. We occasionally use 2 dose or even 1 dose test when we have a very large amount of the compounds. For example, like the compound from the machine learning campaign.
Great. And a reminder to our audience that you can download related resources in the tab to the left of your screen, and this includes a certificate of attendance. So we'll move on to our next question. And during the presentation, you mentioned the XChem library is 100 billion. Do you screen all of these molecules in a single reaction, i.e. in a single tube, or do you split the library into sets of different screening reactions?
That is a wonderful question. So we do indeed screen the entirety of our library, all 100 billion compounds in a single Eppendorf tube. And the reason why this is possible is because we are identifying the hits from the screen using deep sequencing of the DNA tags. In a typical deep sequencing experiment, we'll often sequence between millions, if not billions, of unique sequencing reads. And so this allows us to interrogate the entirety of the library pool in one next generation sequencing run. With that being said, because we're able to perform an entire screen in a single tube, often what we do is we multiplex our different screening conditions. So we will include our target of interest as well as any off targets that may be of interest for that project as well. So in the case of, say, a kinase target, if we wanted good selectivity towards a particular kinase of interest, we would often add other kinases in separate tubes to perform parallel selections and start to directly get mechanism of action information straight from the DEL screen. One point to make about DNA-encoded library hit finding is that it is an affinity-based technique or a binding-based screen. So we aren't able to ascertain whether the hits we find from a DEL screen are functional directly from the selection output. Oftentimes that is assessed in the off-DNA testing workflow that Zoe described during our webinar. But with that being said, by using these different conditions, we can start to interrogate binding sites, selectivity, and affinity within the DEL screen. So that's one of the great advantages of DNA-encoded libraries, is a very, very rapid and easy hit finding on very large compound collections.
Excellent. That was a great answer. Thank you. And so our next question for today is, when using the DEL screening approach, is there a risk that the linker and tagged oligo can impact the target binding of an individual compound? And if the linker oligo does potentially impact the binding, is it possible to have a single compound with linker bound to different positions of the molecule to reduce the impact of, say, position-specific steric hindrance caused by the linker?
Absolutely. So I think the position of the linker and the fact that there is a DNA tag attached to our small molecule is certainly a consideration that we take, not only as we're generating the libraries, but also as we're screening them and analyzing that data. So from a library synthesis perspective, what we often do is we synthesize different versions of the same chemical scheme with that linker and oligo at a different exit vector or position on the molecule. And so this allows us to ensure that the binding events we're seeing in the DEL screen are not driven by a linker or oligo binding event. I will say, on the other hand, there is kind of an attractive feature of having that linker and the DNA tag appended to the library member, and that is to very easily convert the hits that we find from the DEL screen into PROTAC or bifunctional degraders. Oftentimes, when trying to convert a small molecule into a bifunctional degrader, that exit vector or linker point is a very, very critical aspect of designing these molecules. Whereas when hits come from a DNA-encoded library screen, we know a very clear exit vector that already exists within that molecule, and we know what position on that molecule we can modify to best design that degrader. So while sometimes having that linker can be a limitation, we really address that by creating many versions of the library to screen simultaneously, as well as to kind of consider that as we're doing our data analysis. It's a great question.
Great. Well, it looks like that's all we have for today. So I would like to thank our expert speakers again for today's informative discussion and presentation. A big thank you to everyone joining us online. We hope you found this a worthwhile session.